So today we're going to start our study of the circulatory system. Okay? And the first chapter in our textbook on the circulatory system is on the blood. The blood is the medium that the circulatory system uses to transport respiratory gases and nutrients and hormones and wastes and all kinds of things throughout the body. And blood is often associated with life. You know, I've heard it said that some of the rivers in the southwestern part of the United States are the lifeblood of that region because without water, you know, plants can't grow. Without plants, you can't have crops and you can't have wildlife. So, I mean, it's kind of the key to everything. So that's what we're going to start out with today. So, before we start, I want to test your knowledge of blood before we begin to see if some of you have started reading your textbook on chapter 18. So this question is, what's the name for the liquid portion of the blood? We're going to talk about that today. Well, it's just not the cells, but the liquid part of the blood that the cells are suspended in. What's the name for that? Okay. We've got 20 people here counted, so we should have 20 responses. Okay, I think everybody's answered now, so let's take a look. 70% of you said plasma, and that is correct. Plasma is the liquid part of the blood, and we're going to learn today what all the components of plasma contain. All right. So, just to give you a little introduction, the circulatory system includes the heart, which is a central pump. It also includes blood vessels that the heart pumps the blood through to deliver blood to the body, and then the blood itself. Okay? And the circulatory system, as I said on Monday, it is a major organ system, and so actually there's a chapter on blood, which is the one, chapter 18, we're going to start today. There's also one on the heart. There's one on blood pressure and blood vessels. So there's several different chapters that we're going to go through on the circulatory system. But today we're going to talk specifically about blood. So another name for the circulatory system that's sometimes used is the cardiovascular system. Cardiovascular comes from the word cardia, which means heart, and vascular, which refers to the blood vessels. So technically, it's a little more narrow focus than the word circulatory system. Cardiovascular system refers specifically to the heart and the blood vessels. However, it is sometimes used interchangeably with circulatory system. And the study of blood itself is called hematology. So a person who studies blood would be called a hematologist. So blood, one of the primary functions of blood is transport of substances in the body. The circulatory system is the main transportation system of the body. There are a lot of important substances that are transported by blood. One set of substances are the respiratory gases, which includes both oxygen and carbon dioxide. Later on this semester, when we get into the respiratory system, we'll talk about the mechanisms by which oxygen and carbon dioxide are transported. Carbon dioxide is transported a little more complicated There's 3 different ways that CO2 is transported in the blood. Something else I mentioned a little while ago is nutrients. Nutrients like glucose and amino acids and fatty acids and other macromolecules that our body can use to synthesize ATP, which is the form of chemical energy that all of our cells use in our body. And then another thing that our blood transports are metabolic wastes. Carbon dioxide is actually a metabolic waste, but that's also considered respiratory gas. But the most important metabolic wastes are nitrogenous wastes, such as urea and uric acid, because those are toxic to our body, and they're produced by the breakdown of amino acids, which come from proteins. And it's important that we get rid of those wastes. So our circulatory system transports urea and uric acid to our kidneys, which excrete those substances into our urine, which is expelled as urine. Another substance that's transported are hormones. Hormones are chemical messengers that are produced by ductless glands. They secrete these chemicals directly into the bloodstream, so they circulate through the cardiovascular system, and then they're taken up by cells, and it produces a physiological change in cells. So the circulatory system plays a role in the endocrine system, because the endocrine glands produce the hormones. And then also cells of the immune system. Cells like macrophages and lymphocytes and T cells and B cells, those are all transported by the circulatory system to the areas of the body that need those cells, like an infected area of the body or a damaged area of the body. So transport is an important function of the circulatory system. Another important function is related to protection. Blood also contains white blood cells, and some of the white blood cells protect us from infection. Some of them cause inflammation, which is actually a way that can help our body to fight disease. And so white blood cells are part of our defense systems of our body. And as I said, it plays a role in inflammation. Inflammation is a local reaction in a part of our body that causes swelling, which increases permeability of blood vessels, so those white blood cells can get into an area of our body that's damaged. It also raises the temperature, which speeds up the chemical reactions and the healing process. Another function is that blood also transports a protein called complement. Have any of you in here heard of complement? If you've had micro, you've probably heard of complement. Some of you probably are taking micro right now, aren't you? Yeah, you'll probably hear about complement towards the very end of the semester, because it's part of our body's non-specific defenses. Complement is made up of a large number of proteins. I think there's 9 different complement proteins. And they can bind to bacteria and either neutralize the bacteria so they can't cause an infection, or it actually marks them for destruction by cells of the immune system. Antibodies is another protein. Antibodies are immunoglobulins, are proteins produced by cells of our immune system. They don't directly kill microorganisms, but they can bind to them, and it can make it easier for phagocytic cells to phagocytize them. So it can make it easier for a neutrophil or a macrophage to phagocytize either a virus or a bacterial cell. And then blood also contains platelets and clotting factors that can prevent blood loss. And there's three major mechanisms of blood loss we'll be talking about here in a week or so. Those three major mechanisms are called hemostasis. And those, one mechanism, just to give you a preview, is vasoconstriction. When a blood vessel gets damaged, causes the smooth muscles in the walls of the blood vessels to contract. That causes vasoconstriction. That reduces blood loss from a torn blood vessel. Second one is a platelet plug. When platelets get sticky, they form a plug. It's like putting a cork in a hose. And then the third one is blood clotting. Blood clotting involves the formation of a protein called fibrin, which becomes cross-linked, and it sort of forms a net that catches the blood cells and platelets and prevents blood loss. And then finally, another important role of the circulatory system that probably not the first one that comes to mind is regulation. The circulatory system plays an important role in stabilizing the fluid distribution of the body. And it can do this by either taking up fluid, excess fluid from the interstitial spaces between cells, or it can release fluid into areas that are getting dehydrated. And then another way in which the circulatory system regulates things, it can also act as a buffer to prevent changes in the pH of the body fluids. So it helps to buffer acid-base imbalances. And our blood vessels that are close to the surface of our body can either, blood can increase its flow through blood vessels close to the surface in order to dissipate heat, or those blood vessels can become constricted and blood flow can be reduced to the surface of our body, thereby decreasing the amount of heat loss. So regulation of temperature, thermoregulation, is another important function of the circulatory system. So circulatory system is mainly a transportation system, but it also helps to protect us, and it also regulates fluid and pH and also thermoregulation. Okay, so let's talk about the composition of blood. Okay, if you take a sample of blood and put it in a capillary tube, it's just a very narrow diameter tube, just clear. If you fill the capillary tube and then plug one in and put it into a special centrifuge called a hematocrit centrifuge, you can separate blood into two main parts. These two parts are the formed elements and plasma. The formed elements are made up of the blood cells and the platelets. The plasma is a fluid that's made up mostly of water. It's about 90% water. There's also about 6% of the volume of plasma is made up of protein, and about 1% electrolytes like sodium and potassium. Okay. So the concentration of protein in blood is really important because the amount of protein in blood affects the osmolarity of blood. And if your osmolarity becomes too high, then that will draw water out of the different water compartments of the body into the blood. And that's going to increase blood volume, which could increase blood pressure. Okay, so you don't want the concentration of protein in blood to get too high. You don't want it to get too low either. If the concentration of protein in blood gets too low, that decreases the osmolarity, so the blood doesn't absorb as much water. And that can actually result in fluid accumulation in tissues. It can cause edema. Okay? So the most common protein in blood is called albumin. And about 60% of all the protein in blood is albumin. And that acts as a pH buffer. And if your body is producing too much in the way of acids, it can bind to the hydrogen ions and neutralize the acids, or it can also even neutralize bases. And probably most importantly, albumin contributes to a majority of the osmolarity of the blood. So it's important that you have the proper amount of albumin in the blood. Some of the other proteins include antibodies or immunoglobulins, or just simply globulins. Those are proteins that can bind to pathogens and mark them for destruction. That makes up about 36% of the plasma proteins. And then fibrinogen. Fibrinogen is a precursor to fibrin. Fibrin is a protein that forms blood clots. So, but you don't want blood clots forming all the time, and so fibrin is found in an inactive form called fibrinogen, and that makes up about 4% of the protein in plasma. So if we add that all up, what do we get? Let's see, 60 and 36, that's 96, and 4, that's 100%. Okay. Now there are probably a lot of other proteins in blood as well, but they're found in very, very small concentrations. Okay, so these numbers are just probably rounded up. The other major part, as I mentioned, are the formed elements. This is the part that settles out in the bottom of a hematocrit tube if you centrifuge it. Formed elements include erythrocytes, which are the red blood cells that we're going to learn about in lab this week. Leukocytes, which are commonly called white blood cells, and platelets. Platelets are cellular fragments that are produced in the red bone marrow. Erythrocytes are the most numerous, and they form, on average, it forms slightly less than half of the volume of whole blood, the red blood cells do. Typically, males have a slightly higher red blood cell count than females on average. It's probably as a result of hormonal differences. Leukocytes, on the other hand, which are about 1,000 times less numerous than red blood cells, they only make up less than 1% of the total blood volume. Typically, there's 5 to 10,000 white blood cells per cubic millimeter of blood, as opposed to about 1 million red blood cells per cubic millimeter. And then platelets also make up much less than 1% of the total blood volume, even though they're much more numerous than white blood cells, but they're so much smaller that they don't make up much of the volume. So there's some general properties of the blood that are very important that affect how well blood can function as a transport medium and how well it can function in regulation and protection. The first general property is viscosity. Viscosity is the thickness of blood or the stickiness of blood. And that's affected by the relative amounts of water versus proteins, basically. So the more water, the higher the proportion of water in the blood, the lower the viscosity. A low viscosity fluid is very watery. It flows very, very easily. If you have a lower proportion of water, higher proportion of protein, then the blood is going to get thicker. and it's going to flow more sluggish. And that can have a problem, and this can happen just as a result of dehydration. If you lose too much water and don't take in enough water in drinking. Too high of a viscosity can make it harder for the heart to pump the blood, so it can put strain on the heart, and it can slow down the flow of blood through the blood vessels because it's so thick. And if blood flow is slowed down, then that increases the chance that you'll get blood clots forming in blood vessels. And there are a number of problems with blood clots. One of them, it can block the flow of blood to vital organs, and it can cause organ failure. And another problem that's even more serious is that if a large blood clot breaks loose, and then it circulates through the body, it can get stuck in the lungs, and that can block flow of blood to the lungs, so your blood's not going to get oxygenated. So that's called a pulmonary thrombosis. You can literally suffocate because of that, because your blood doesn't get oxygenated. Too low of viscosity can cause blood to flow too easily, and it also can cause a lot of water to leak out of the blood vessels, and it can cause edema or swelling. Another important property is osmolarity. Osmolarity is the molar concentration of dissolved particles in per liter. And osmolarity is important because osmolarity affects the movement of water. So if the osmolarity of the blood gets too high, then it's going to draw water out of the interstitial fluid into the blood vessels. That will increase blood volume, and that could cause high blood pressure. If the osmolarity of blood too low, then water is going to leak out of blood vessels, and it's going to cause fluid accumulation or edema. And the osmolarity is affected primarily by the amount of protein that's in blood, as well as the blood cells. So blood cells also, blood cells and protein produce what's called colloid osmolarity. And that it's just increase in osmolarity due to particles that aren't dissolved. Now, one example of a problem that can occur due to an increase in osmolarity of the blood is a condition called hypoproteinemia, which is also known as kwashiorkor. Hypoproteinemia is a form of malnutrition. And it's a form of malnutrition where a person's not getting enough protein in their diet. And you can get this condition from eating a diet that's high in corn, because corn has carbohydrates in it. Corn does not have a real high concentration of protein, though. So if you subsist on a diet of mostly corn, you could become protein deficient. Now, if you're not getting enough protein in your body, in your diet, then your body can start breaking down the albumin that's in your blood and use those amino acids for synthesis of other proteins. That's going to decrease the osmolarity of your blood. And if your osmolarity of blood decreases, then your blood is not going to absorb as much water from your extracellular fluid, like your interstitial fluid. And so fluid's going to accumulate. And so that's why you see sometimes pictures like this of starving children, but their bellies look swollen. that's referred to as ascites. Ascites is fluid accumulation in the abdomen. And that's actually a sign of malnutrition. Okay, it's not that they're stuffed with food, it's that there's fluid accumulating because their blood doesn't have enough protein, so their blood can't absorb water from their extracellular fluid. So the fluid accumulates. So it's important that blood maintain an optimal osmolarity. The most important contribution to osmolarity is called the colloid osmotic pressure, and that's the contribution, mainly the contribution of protein in the blood. Concentration of sodium and also erythrocytes also play important roles. Okay, so now that we've kind of got an overview of blood concentration, let's start looking at some of the individual components of the formed elements. This is the solid part of blood. So the first one are erythrocytes. Erythrocytes are the most numerous of the formed elements. And erythrocytes are very, very simple in structure, actually. They're just really a bag of cytoplasm, and they have very few, if any, organelles. They're shaped like a biconcave disc. You can see the shape here. And during the development of red blood cells in the red bone marrow, they lose their nucleus and most of their other organelles. So they lack mitochondria, they lack a nucleus, they lack most of the other organelles, and they can get by without mitochondria because they rely mostly on just glycolysis for their energy production. That's a form of anaerobic metabolism. And mainly all they contain is a protein called hemoglobin, which we'll take a look at here in just a second. So the main functions of red blood cells are the transport of oxygen from the lungs to the tissues, and also the transport of carbon dioxide from cells and tissues to the lungs, where the carbon dioxide can be exhaled. So this is a model over here in the upper right of a single hemoglobin molecule. The hemoglobin molecule consists of 4 different subunits, or 4 different polypeptides that are all linked together. And in the center of each subunit is an iron-containing compound called a heme group. So this is a heme group down here, and you can see these little red structures up here. Those are the heme groups. So there's one heme group in each one of the subunits of hemoglobin. There's actually 2 different kinds of subunits in hemoglobin. There are there's 2 alpha subunits, and there's 2 beta subunits. They both do the same thing. They both contain a heme group. They both are involved in transport of oxygen. They just vary slightly in their amino acid sequence. So there's 4 globin units or subunits in the hemoglobin molecule. Each one of those 4 has 4 heme groups. And heme is the part of the hemoglobin molecule that oxygen binds to when oxygen is transported in the blood. So since there are 4 heme groups in hemoglobin, each hemoglobin molecule can transport up to 4 oxygen molecules. Okay, can transport anywhere from 0 to 4, okay, because there's 4 different heme groups. So this heme group is the main reason that you need at least some iron in your diet, because you need iron to synthesize heme in order to make hemoglobin. If you don't have enough iron in your diet, then your blood can be deficient in hemoglobin, and that can cause anemia, which decreases the ability of your blood to transport oxygen. So like I said, oxygen is transported by binding to this heme group. About 5 to 10% of carbon dioxide is transported by binding to the globin portion, which is the protein portion of hemoglobin. When you add CO2 to the globin portion of hemoglobin, that forms a compound that is called carbaminohemoglobin. Okay? When hemoglobin is carrying oxygen, it's called oxyhemoglobin. And when it's not carrying oxygen, it's called deoxyhemoglobin. We'll be talking more about that when we get to the respiratory system. Okay, so the carrying capacity of your blood is mainly affected by the number of red blood cells and the hemoglobin concentration. So one test that's often done clinically to determine if you have a good oxygen carrying capacity in your blood is to do a hematocrit test. To do a hematocrit test, what you do is you take a very narrow diameter hollow tube, glass tube, and it has such a narrow diameter that if you have a drop of blood, you can put the end of the capillary tube in it and it will just draw up the blood by capillary action. So you fill up a capillary tube, and then you plug one in, and then you place it into a special centrifuge for doing hematocrits. It's called a hematocrit centrifuge. You want to make sure you put the plug in to the outside of the centrifuge. Otherwise, all the blood will run out of it. Because normally, one end is plugged, the other end is still open. And if you put it in the wrong way, the blood will go all over and make a big mess. But anyway, you put it in the centrifuge, and then you let it spin in the centrifuge for about 4 minutes, and that will separate the formed elements from the plasma. And the red part of the hematocrit tube after you centrifuge it, that's all of the formed elements. So that includes the erythrocytes, leukocytes, and the thrombocytes. But it has a reddish appearance because the erythrocytes make up such a high proportion of the volume of the formed elements. So actually, the volume of the white blood cells and the platelets in this hard packed volume is really insignificant. You don't need to worry about that. So by measuring the length of the tube occupied by the red blood cells and dividing that length by the total length that was filled with whole blood and converting that to a percentage, that gives you the proportion of whole blood that's made up of the red blood cells. And the normal range of values for a hematocrit for women is 37 to 48%. Slightly higher than men, because men usually have more red blood cells than women. So for men, normal range is a little higher, 42 to 52%. So on average, though, if you average everybody together, both men and women, usually the proportion of blood made up of red blood cells is slightly less than half, slightly less than 50%. Some highly conditioned endurance athletes may have hematocrit, though, more than 50%, but that's a small proportion of the population. So if your hematocrit is lower than normal, lower than those normal ranges, then that means that you're anemic. You have anemia, and that's going to decrease the ability of your blood to transport oxygen. So you're not going to have as much endurance. You'll fatigue easily if you're anemic, and that can be caused by a large number of different reasons. Well, we're going to go over that next week in lab. one of them is not having enough iron in your diet. That can cause anemia. Now, even if you have a normal hematocrit, you can still be anemic though. There's two ways that you can be anemic. You can be anemic because you don't have enough red blood cells, or you can be anemic because you have a normal number of red blood cells, but you don't have enough hemoglobin in those cells. So there's another test you have to do, and this is one of the things we're going to do in lab next week. You can do a hemoglobin test. You can test for the concentration of hemoglobin in the blood. And we have some small meters that look like this. This is the same meter that we're going to use. These meters, though, are no longer being made. So since the meters are no longer being made, they're no longer making these sample cards. They're no longer available. And so I'm going to have to replace those meters probably no later than next year because we won't be able to use them without the sample cards. And I'm kind of running low on those, but we probably have enough for this year. So anyway, to test, there's several different types of hemoglobin meters. This is a little handheld one. It's called a point of care meter. There's a little plastic card, and you insert in a little slot on one end of the meter. You turn it on, and after it turns on, you place a drop of blood. There's a teeny tiny hole right in the center of this larger depression. There's a smaller depression and a larger depression. You got to put that blood right in that very center well. And you need to make sure you cover the whole bottom of that well, otherwise it won't work. You'll get an error reading. So if you put the drop of blood in there, then the meter starts counting down. It takes 90 seconds to get your results. Some of the newer ones are a lot faster. That's probably why they're not making these anymore, because these are too slow. Because a minute and a half is too slow. So you put the drop of blood in there, and then after it's done, it will tell you the concentration of hemoglobin in your blood. So since men have more red blood cells than women on average, they typically have a little higher concentration of hemoglobin because there's more blood cells that have hemoglobin. So for men, the normal range is 13 to 18 grams per deciliter of hemoglobin. For women, this range is slightly lower, 12 to 16. So if your hemoglobin concentration is lower than normal, that's also an indication of anemia. Okay. So you have to do both tests to rule out anemia. If somebody's tired all the time, they think they might be anemic, you have to test both their hemoglobin and you have to do the hematocrit. All right. Any questions? Okay. Have any of you in here ever given blood? Okay. Quite a few of you had. Looks like half or more. When you give blood, they usually test your hemoglobin to make sure that you're not anemic. Because if you're anemic and they remove, a unit of blood, then you're going to have that much less hemoglobin in your bloodstream, and that can make it even worse. So they sometimes have like a little desktop hemoglobin meter. It's got a little cuvette, and they fill the cuvette with blood, and they stick it in there, and it tells you what's your hemoglobin concentration. Long time ago, in fact, some places still might do this. Another way that they measured hemoglobin was they had a solution. It was sort of a clear blue solution. And the solution had a certain osmolarity. And what they would do is they would put a drop of blood in that fluid. And if the drop of blood sank to the bottom of the container of fluid, that meant that your hemoglobin concentration was good. If it didn't sink, that meant that you didn't have enough hemoglobin in your blood and that you're anemic, and then they wouldn't let you give blood. So that's really kind of a low-tech method of doing it, but they have higher-tech methods now. Okay, so the reason that the hematocrit and hemoglobin concentrations are higher in men than women are because androgens, which are the male hormones such as testosterone, stimulate red blood cell production. Okay? We don't know why, but it just does. And men have higher concentration of androgens than women. Also, women of reproductive age lose blood every month through menstruation. And because they lose blood every month, that decreases the number of red blood cells. The fewer red blood cells you have, the less hemoglobin you have. And also, hematocrit is inversely proportional to body fat. So if a person has higher body fat, they're going to have lower hematocrit. And on average, there's a lot of individual variation, but on average, women of reproductive age have a higher body fat proportion than men. So theoretically, if you're a female, you could boost your hematocrit just by losing weight, just by losing body fat. Okay. All right, let's do another review question. Okay, so this is a question that has to do with the structure and function of hemoglobin. So if you have a total of 100 hemoglobin molecules, how many oxygen molecules could those 100 hemoglobin molecules carry? Still have one more person, Luke and Dan, an answer. Oh, we have one person left. Okay, so everybody's answered. Sorry about that. Okay, so most of you said 400, and that would be correct because each hemoglobin molecule has 4 heme groups. Each heme group can bind to one oxygen molecule, one O2 molecule. So 100 hemoglobin molecules can transport 100 times 4 or 400 oxygen molecules. Okay, I think this would probably be a good place to stop because we don't have really the time to get into the next section of this chapter. So why don't we stop here and we'll continue to talk about some other aspects of blood on Friday.